wearing Cuban links, designer mix, Inglewood's finest shoes. Don't look too hard, might hurt yourself. Known to give the color red the blues. Woo, shit. I'm a dangerous man with some money in my pocket. Keep up. So many pretty girls around me and they're waking up the rocket. Keep up. Why you mad? Fix your face. Ain't my fault they all be jocking. Keep up. Players only. Come on, put your pinky rings up to the moon. Girls, what y'all trying to do? 24 karat magic in the air. Head to toe so clear. Look out. Everywhere I go, they be like, oh, so bad. Everywhere I go, they be like, oh, so bad. Everywhere I go, they be like, oh, so bad. Now, watch me break it down like.
Integral membrane protein, that also known as the IMP. It is located at cellular membrane. Yes. Okay, it has three functions, which is first is the cell-cell communication. Second one is the adhesion cell, and the last one is the cell signaling. So there is three types of IMP. First one is the transmembrane protein, which is expand the membrane either by the single or multiple pass. And the second one is the cytosolic membrane associated protein, and the last one is the lipid link protein that covalently with the lipid. Okay, there is three types. There is three steps for the purification and activation process. First one is the extraction by using detergent buffer, and the protein separation can be done by centrifugation, and the protein identification can be done by using the LC-MS process. So there is three factors that can cause the precipitation for this protein. First one is the ion strength, temperature, and also the salt concentration. So in order to study about this IMP protein, we choose the mouse liver microscope, which is the main source that is rich with this IMP protein.
The microsome is the physical light activated reform from the endoplasmic reticulum. So as first, the sample is the mouse liver tissue. It will undergo the dissection and homogenize using the homogenizers machine. Become the homogenate. The homogenate will subject to the centrifugation, and we will obtain the pellet and the supernatant as it. At the end of centrifugation, the pellet consists of the nucleic acids, large organelles like mitochondria will be discarded. So we want the supernatant to subject to the next round of the centrifugation. The supernatant will undergo centrifugation at high speed, and to obtain the pellet is consists of microsome crude sample. The microsome crude sample in the pellet will be washed with the sodium carbonate to remove the peripheral associated protein. So at the end of this preparation step, we will obtain the microsome fraction.
So continue from what Jimmy has got just now, it is the microsome fraction, which is washed with the sodium carbonate, and then this fraction will be dissolved and resuspented in the aqueous 2% of the treated X114, and then it will be incubated on ice for half an hour with the frequent vortexing during the interval of incubation. Then after that, this suspension will be centrifuged at high speed and at low temperature for another 20 minutes to get the supernatant and also the pellet. As for the supernatant, it will be incubated for another 15 minutes at 37 degrees Celsius, and this process is important to achieve the phase participation. Then after this mixture will be centrifuged again for another 30 minutes, so that you'll get a two-phase layer, which is the aqueous phase and also the detergent phase. And both of these phases can be further purified to repeat the phase participation process. And then this is the method to repeat the phase participation process of these two phases. Then after this, the protein collected in the aqueous phase and also the detergent phase and also the pellet will be collected and harvested by the acetone precipitation. After that, the protein will be air dried and stored in the negative degree, 80 degrees Celsius freezer.
In this study, they use viral protein concentration assay performed to estimate the protein concentration in microsome samples. First of all, let's see what is viral protein concentration assay. It is based on Bradford. It's a simple and accurate procedure for determining the concentration of solubilized protein. And it is a dye binding assay, which differential color change of a dye occur in response to various protein concentration. The response of dye to protein solution at 465 nanometer to 595 nanometer with binding to protein occur. So let's see the procedure how they did. There are 5 concentration of BSA standard was prepared to generate a standard curve, and then equal volume of BSA standard and unknown sample were mixed to 3 microliter, which is mixed with the 100 microliter of Biuret dye reagent, which is 5 times diluted, into 96 well plates. The samples were triplicated. And the absorbers at 595 nanometer was measured to determine the protein concentration of microsome samples. And that's it from me.
SBS fish is sodium dolichite sulfate polyacrylamide gel electrophoresis, which is commonly used for the separation of proteins based on their molecular weight and based on their differential rate of migration through a sieving matrix under the influence of an applied electrical field. Based on our reference, so I will explain detail about the method of SBS. For the first one is Aliquot 30 microliter of membrane fraction and then resuspended in the new NUPH LDS sample buffer and then boiled at 95 degrees for 10 minutes and then loaded into the 4 to 12% NUPH Novak by freeze gel, and then after that the electrophoresis will be performed at 150 volt, which is constant voltage, in the MDS running buffer, and then the last one is the protein will be visualized by using infrared processing to pieces. What is in-gel protein digestion? This particular method is done in order to cleave the protein that present in the polyacrylamide matrix in the gel before. That's before this, they're doing the SDS stage. Okay, the first step for this particular step is to excise the gel banks from the gel into 2 or 3 millimeter gel thickness, and then those gel slices are de-stained by using ammonium bicarbonate in they were immersed in the ammonium bicarbonate for about 30 minutes at 30 degrees Celsius to remove the corrosive blue stain. And then those de-stained gel slices will be reduced with 10 millimolar of DTT for 30 minutes. And this is in order to reduce the sulfide bond that present within the proteins. After the reduction step, the gel slicer will be then alkylated with 25 millimolar iodoacetic acid for 20 minutes. This iodoacetic acid is used in order to prevent the disulfide bonds from reforming, because it will react with the thiol groups in the amino acids. Next, the gel slicers are trypsinized by using trypsin for 5 hours at 37 degrees Celsius. This trypsin is an enzyme which will slice the protein into peptides at amino acid lysine or arginine. And those gel peptides are then extracted and dried by centrifugal lyophilization, and those digests will be subjected to the next step, which is mass spectrophotometry analysis, which will be explained by my colleague, Roslina. LCMS means liquid chromatography mass spectrometry analysis. This purification and identification of integral membrane proteins. Protein identification by LCMS is one of the important steps rather than extract from native membrane to detergent buffer and also protein separation. Protein identification by LCMS is the third important step. Why it is needed? Because membrane protein have very low expression level than cytosolic membrane, and also limitation of protein abundance. In this particular research, LC-MS-MS characterization have been done by combining the TX114 phase separation and gel LC-MS-MS, which revealed 146 proteins more. Okay, now I'll explain about how in detail how the nano LC-MS-MS analysis have been carried out. The 96-well plate have been inserted with digested peptide, and then it will be loaded into the autosampler for injection, and then fractionation by nano flow reverse phase HPLC. If you guys can see, the nano flow reverse phase HPLC looks like that. And then the positive ion mode from the HPLC is noted for data dependent acquisition, and then the mass spectroscopy scan being set its resolution to 30,000 RPM, where each scan will be recalibrated by co-injecting the minimal standard from ambient air. And then 5 moles intense ion per cycle will be fragmented and analyzed in linear track. And then the targeted ion will be selected and excluded for 180 seconds. And then this step will be proceed to protein identification and bioinformatic analysis. This membrane protein is important in Human Genome Project. So there is a limitation to isolate and purify this membrane protein, which is it has low copy number, so it is hard to obtain the membrane protein. The second one is because its hydrophobic nature, and the third one is low expression level of this protein. Because of this, it will create unfractionated mixture, which difficult to isolate this protein. So the last one is issues to choose the protein separation technique because of the limitation that I've mentioned before. So that's all for me.
Firstly, it is utilized the sucrose and iodine in density gradient certification, and it also use hydrophilic biotinylation and use antibody for the affinity. Next. So there's an advantage and disadvantage for using this phase separation. Firstly, the protocol is simple, but yet it is only used a few detergent and for the membrane protein combination. Secondly, it is compatible with most chromatographic method, but it is high concentration in the detergent use. And thirdly, membrane protein can be simultaneously purified and concentrated, but it is not widely used for proteomic because not many researchers doing this. Next. So for the conclusion, TX114 phase fractioning yields more number of protein compared to the unfractionated MF, which is microsome fractionate. And that's all from us. Okay, I will call Ekazanaria and Punita for the question, and I will call Z for the reward.
For you, for those who answer the questions, okay? Okay, let's go for our first question.
Okay, so the first question is mention 2 types of integral membrane protein. Anyone can answer?
You don't answer.
Kevin.
protein, cytosolic membrane associated protein, and another one is lipid link protein. Okay, no chocolates. So the second question is bio-red protein assay is based on dash and known as dash assay. So what are the answers? Anyone want to answer the question? Try. It's Bradford. And known as diaminoacid. Yes, correct.
Okey, nak kena jawab dulu ni.
Okay, let's proceed with the question.
So the question is what types of mouse organelle used in this experiment?
Cadbury guys, Cadbury.
guys. It's Cadbury guys.
Cadbury guys.
Is it a liver mouse cell?
The specific name. We had repeated the name few times. Okay, are you? Correct. Okay. Fourth question. Mention one advantage of Triton X100.
Single to use, compatible with most chromatographic method.
Yes, I want the big one. Saya mau yang besar sikit. Ini apa ni?
The last question. So, where is the location of this IMP protein?
Not in the slide. It's not in the slide. Yes, it is. I mean, the question is not in the slide.
In this slide.
In location, right? Oh, cellular memory.
The location for this IMP protein.
Kevin. Okay, so korang buat apa hari ni?
I managed to answer the question and I got chocolate. Kau dah dapat chocolate.
Hi. So what are we presenting today?
We are going to apa nama dia?
Membrane protein.
Membrane protein.
Okay, good. Stay focused. Hi, sister. Okay, guys. What are you doing? Please stay focused during the class. What? Hi. Go. Kak Mira, time ni kau nak makan kan? Say hi guys. Okay, good. Sister, jangan main phone, ya Allah. Sebaik video ni, aku nak tunjuk benda ni. So, hi. So, what are we doing in the class for today?
So for now, we are watching the presentation from group 5 that present the protein membrane.
This is the group 5. Bonita is presenting. Location of IEP.
It's too late to apologize. It's too late.
Yay! Luna. 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 Say hi.
Okay, 1, 2, 3.
Thank you for watching. Bye.